let's have a go at a biology past paper. So this is a paper 1 higher tier from the Trilogy series of AQA. Now the link for downloading the paper is down below. I suggest you print out the paper first and have a go for yourselves. In this first question, we're going to be looking at cells. It says draw one line from each cell to the type of cell where the structure is found. Now, if we have a cell that has a nucleus, then we would have a eukaryotic cell. So that matches down to this one at the bottom. And alternatively, when we have cells that don't have a nucleus like plasmids, they are the prokaryotic cells. And then a cell which has a permanent vacuole would be our plant cell because that's where the cell sap is. So continuing with cells and cell structures, we've got to correctly identify structures A, B and C. Now if we look at structure A, it's the middle part of a plant cell, so it's going to be the vacuole. And if we look at part B is the place where proteins are synthesized, and that would be the ribosome. So it looks like this is going to be our correct answer here. Let's just make sure the outside of a plant cell is our cell wall. So our answer would be this one here. In this question, a student observed slides of onion cells using a microscope. So we have two slides, slide A and slide B, and the slides are not clear to see. So we've got to describe firstly for slide A what we can do to make that more clear. So because that just needs a little bit of focusing, you would only be adjusting the fine focusing knob for slide A. Now for slide B is requiring a lot more magnification, so we need a more powerful lens, and we'll get that by adjusting the objective lens. Now for this next question, the student made the necessary adjustments to get a clear image. And figure 3 shows the student's drawing of one of the cells. So the real length of the cell was 280 micrometers, and in the diagram we can see we have 112 millimeters. So we need to calculate the magnification. So the magnification, I'm going to use our equation, is the size of the image divided by the real size of the image. Now, first of all, what we need to do is we need to make sure that the real size and the actual drawn size of the image are in the same units. So if I'm going to change 112 millimeters into micrometers, I need to multiply it by 1000. So using that in my equation, that means I would have 112,000 micrometers as the image size, which was measured, divided by 280, which is the real size, to get my magnification. And if I calculate that out, the magnification is 400 times. In this question, we're told that coronary heart disease, CHD, is a non-communicable disease. CHD is caused when fatty material builds up in the coronary arteries. Explain what a non-communicable disease is. So here we've just got to explain that with a non-communicable disease, that a pathogen is not able to pass on the disease, so it's not spread from person to person. Now, in the next question, we're shown a diagram, figure 4, of the arteries with the fatty material built up which is reducing the volume of blood that would be circulated. Now, our question is to explain how CHD can cause a heart attack. So first of all, we need to state that the volume of blood is reduced due to the fatty material, and if the volume of blood circulating to the heart is reduced, then that means there's going to be less oxygen going to the heart, and if less oxygen is going to the heart muscles, that means that there would be less respiration happening in those muscles. In this question, we need to explain how lifestyle and medical risk factors increase the chance of developing CHD, and it's a 6-mark question, so we need to be linking lifestyles and the medical risk factors together. So examples of the things that we could say are, first of all, we could talk about lack of exercise, can lead to obesity, high cholesterol, and diabetes. We can also talk about a poor diet, can also lead to obesity, high cholesterol, diabetes and blood pressure. We can talk about smoking, which can lead to increased blood pressure, as well as buildup of cholesterol. And we could also maybe talk about if you consume too much salt, can lead to high blood pressure. So for this question, as long as you're saying what it is that you're doing, which can lead to the impact that it's having on your body. In this question, we are looking at photosynthesis, and first of all, we've got to say which is the correct balanced equation. So photosynthesis is carbon dioxide plus water produces glucose and oxygen. So the only equation which has carbon dioxide and water as the reactants would be this one. Then for our next question, we're asked what type of reaction is photosynthesis. Now, photosynthesis requires energy from the sun for it to happen. So if we're taking in energy, that would be an endothermic reaction. Now, in this question, a student investigated the effect of light intensity on the rate of reaction of photosynthesis. So we have our light shining on the pondweed, and they're counting the number of bubbles produced. The plant is placed 5 centimeters from the light source. They're measuring the rate of photosynthesis by counting the number of bubbles in 1 minute, and they're repeating with the pondweed at 10 centimeters, 20 centimeters from the light source. So counting the number of bubbles in 1 minute is not an accurate way to measure the rate of photosynthesis. And we've got to make 2 suggestions how this method could be improved. So first of all, if you're counting the number of bubbles, instead of counting the number of bubbles, what we could do is we could measure the volume of gas that's being produced. And then the second way is, instead of just looking at 1 minute, we could maybe increase the length of time that we're doing this for. So it could be a longer period of time. For this question, we're told that an LED light source does not get hot, and that's what's used in our investigation. Explain why it's important that the pondweed remains at constant temperature. Well, the first reason has to be that if we used a light source that increased the temperature would affect the rate of photosynthesis. Also, if the pondweed gets too hot, the enzymes that are in the pondweed would be denatured. And if the enzymes become denatured, they'll stop working. Now, for the next question, light intensity can be calculated, and we're given an equation where I is light intensity and D is the distance from the of the pondweed from the light source. So the student used these distances. Explain how light intensity changes as the distance of the pondweed from the light source is doubled. So what we can do is we can choose two of the distances where one of the distances doubled, and we can see, using the equation, we can see the impact that it had. So if we say at 5 centimetres, we would have, using the equation, 1 over d squared, we would get 1 over 5 squared, and that would give us 0.04. And then if we double the distance, so at 10 centimetres, still using 1 over d squared, we would have 1 over 10 squared, and that would give us 0.01. So doubling the distance, light intensity is, and if we look, doubling the distance, light intensity is quartered. Okay, for this question now, we've got a results table, and we've got 5, 10 and 20 centimetres from the light source and the number of bubbles produced in 1 minute. So it says predict how many bubbles of gas would be produced in 1 minute if the pondweed was 20 centimetres from the light source. Now, 40 centimetres from the light source. Now, 40 centimetres is double 20 centimetres. And we worked out that if you double the distance, then the light intensity is quartered, so we would expect the number of bubbles to be a quarter. So 40 centimetres is double 20 centimetres, therefore there would be quarter of the bubbles. And so 8 divided by 4 to quarter it gives us 2 bubbles. That's what we would expect. Now for our next question, it says describe how the student could change their method to investigate the effect of carbon dioxide concentration on the rate of reaction. So it's the sodium hydrogen carbonate that's producing the carbon dioxide. So what that therefore means is that we would have to look at having different concentrations of the sodium hydrogen carbonate as our independent variable. And the controls, the things that we would need to keep the same, would be we would have to make sure that the distance from the light source is the same each time we do the experiment. And we know that temperature affects the rate of reaction, so we would need to make sure that the temperature is kept the same. There are other things that we could have as the controls. For example, we could say that we need to use the same plant. We could also say that we need to make sure that the time is the same. A student investigated the effects of different concentrations of sugar solution on pieces of carrot. This is the method used. So they've weighed 5 pieces of carrot. They've placed each piece into a different test tube. Into each test tube, they've added 20 centimetres of water or the sugar solution that's shown in figure 6. They've left it for 2 hours. They've removed the carrots and dried them with a paper towel. They've reweighed the carrots and then they've calculated the percentage change in mass of each piece of carrot. So we have our results table, and our first question is suggest why the student calculated the percentage mass change of each piece of carrot. So our answer to that question would be because the pieces of carrot were not all the same mass at the start. For this question, first of all, we needed to label and choose an axis for our y-axis. So you get 1 mark for choosing a suitable scale. Now I've chosen to go have differences of 5 because that allows me to get my smallest and my largest number on. And the label for the axis is the percentage change in mass. So you have to do both of those things correctly to get 1 mark. Then we have to plot our results, and our accuracy of plotting our points have to be within plus or minus half a square. And we would get 2 marks for plotting all of our points correctly. And then the last mark is to draw our line of best fit. So you use a ruler and you draw your line of best fit. Now for this question, we are being asked to estimate the concentration of sugar solution inside the carrot. And we've got to use our graph from figure 7. So the way that we use our graph is we see where the y-axis is cutting the x-axis when the y-axis is 0. So that means the percentage mass increase or decrease would be 0. And from my graph, it cuts it at a sugar solution of 0.48 moles per decimeters cubed. The next question is asking us to explain why the mass of the carrot at 0.6 moles per decimeters cubed has changed the mass. So here what's happened is the mass of the carrot is decreasing. Okay, now the mass of the carrot has decreased due to loss of water by osmosis, and this happens across the partially permeable membrane because the concentration of the sugar solution on the outside is greater than the concentration of sugar inside the carrot. So therefore, the water moves from inside the carrot to outside the carrot, creating a mass decrease of our carrot. then for our next question, the student repeated the investigation using boiled pieces of carrot. The pieces of carrot did not change in mass. Suggest why. Boiling the carrot would usually damage the cell membrane, and this means that osmosis would no longer be able to happen. For the next part of the question, it says one reason for the decrease in the number of cases of measles is that more children were vaccinated against the disease. Vaccinating a large proportion of the population reduces the spread of the measles virus, and we're asked to explain why. So first of all, if people are becoming vaccinated, then that means that majority of the people are becoming immune, so they don't become ill or become infected. And if less people are becoming infected, there's less chance of them spreading it to people who are not immune or have not had the vaccination. Now, in this question, we are being asked to explain the differences between antibody production after the vaccine injection and after exposure to the measles virus. And we're using figure 8 to help us. Now, when we look at the graph in figure 8, we can see that after the vaccine, there's a small increase in the number of antibodies, the concentration of antibodies in the person's body. However, when there's exposure to the vaccine, due to the memory of the white blood cells have to this particular pathogen, the antibodies will be produced in much larger numbers and much more quickly. In this question, we're going to be looking at stem cells. So it says, give one place in a plant where stem cells are found. So the main places we would say is either in the meristems, but you could also say in the tip of the shoot or the tip of the root. Now for the next question, it says, what is one economic use of the plant stem cell? Now, because it's an economic use, we would usually be looking at increasing numbers of the plant. So although plant stem cells can be used to genetically modify crops, to create new species, or create variation in plants, the key thing for the economic use is to produce large numbers of identical plants. So embryonic stem cells divide by mitosis. Figure 9 represents a cell cycle for a human embryonic stem cell. Now the first question says the mass of DNA in the cell at the start of the cell cycle is 6 picograms. A picogram is 10 to the minus 3 nanograms. Convert 6 picograms to grams. So therefore, if we use this information, so 1 nanogram is 1 times 10 to the minus 9. Therefore, if a picogram is 1,000 times smaller, 10 to the minus 3 times smaller, then that means 1 picogram would be 1 times 10 to the minus 12. So if we're converting 6 picograms to grams, that would be 6 times 10 to the minus 12 grams. Now the time taken for the complete cell cycle is 15 hours. Now we've got to calculate using the pie chart how many hours the cell spent in mitosis. And we've got to give our answer to 3 significant figures. So if we look at our pie chart, we should be able to see that 28 degrees out of 360 degrees is mitosis. Therefore, we would do 28 divided by 360, and we would times it by our time taken for the whole of the cell cycle, which was 15 hours. And if we do that, we should see that the time taken is going to be 1.16 recurring, and because we have to give our answer to 3 significant figures, our answer should be 1.17. We round up because the number after the 6 would be another 6, which therefore makes the answer 1.17. Now for the next question, we have to describe what happens in each of the 3 stages of the cell cycle. In the next part of the question, we are being asked to describe what happens in each of the 3 stages of the cell cycle. So, stage 1 is where we are trying to have cell growth. And this happens by chromosomes replicating. Then in stage 2, one set of each of the chromosomes moves to each end of the cell, and then the nucleus divides. And then in stage 3, this is where cytokinesis happens, which means the cytoplasm and the cell membrane also divides, and we end up with 2 identical cells. In this question, we're being shown embryonic stem cells that are produced in therapeutic cloning for use in patients. So we can see that we have a donated egg cell, and the nucleus is taken out. So then we have the nucleus from the body cell from the patient, which is then used to be cloned. So embryo is produced, and the stem cells are taken from the embryos and cultured, and there's some cells that are not used as well. So we've got to give 2 advantages and 2 disadvantages of the therapeutic cloning in medical treatments. So the first advantage that we could say is that we could use this type of therapeutic cloning to maybe cure medical conditions or cure diseases. The second advantage we could say is that because we're using the body cell from the patient, there's less chance of the treatment being rejected. The disadvantages could be that potential life is destroyed during this process, and also potential for transferring viruses and infections could be at a higher rate. So this brings us to the end of the Biology Higher Paper 1 for Trilogy. I hope you found this useful, and if you would like to see more content like this and see the chemistry past paper as well as the physics past paper, please hit that subscribe button and put your notifications on. Good luck with your learning, and I hope to see you back on my channel soon.